Hello, everybody. So today's going to be notes on meiosis. So I told you a little bit the other day that meiosis is the other cell division, special cell division, because the purpose is to create sex cells, which are also called gametes. Gametes would be sperm and egg cells. So only organisms that undergo sexual reproduction have this type of meiosis. So if they have gametes, egg and sperm, these types of organisms, like us humans, have this cell division called meiosis. Now, you may have noticed more stages in meiosis. There's a meiosis 1 and a meiosis 2. You'll notice the phase names are pretty much similar. The only thing added to that is because there are actually two cell divisions. There's a prophase 1 and then a prophase 2. Okay, so first all the meiosis 1 phases go through the process, and then you have meiosis 2, which has prophase 2, metaphase 2, anaphase 2, and telophase 2. Meiosis takes place in the sex organs. Now, there's going to be some new terminology here that we're going to introduce that you're going to be using as far as even going into genetics, which is our next unit. So homologous is one of those terms. So first question is, what are homologous chromosomes? So they're the pair of the same chromosome. One is inherited from mom, and the other is inherited from dad. So in the picture here, all right, we have this purple one and this green one. This is considered a homologous pair. One came from mom, one came from dad. On the left here, this is what a chromosome looks like before interphase. And this is after the replication has taken place. You probably remember the term sister chromatid and the term centromere, which holds those sister chromatids together. So looking a little closer at the homologous chromosome. So keep in mind, one came from mom, one came from dad. So what is different about them? I mean, they are the same in the sense that they contain the same genes. But what is different is they may have different alleles. All right, go ahead and pause if you need to catch up. So what are alleles? So you want to separate alleles from a gene. They're not the same. An allele is a different variation of the gene. Example, you have a gene that controls eye color. An allele would be the different possible options of that eye color, such as brown eyes. So mom might have the allele for brown eyes. Dad may have an allele for blue eyes. Okay, and that goes with all characteristics that are found on whichever chromosome. And remember, we have 23 pairs or 46, right? So they're paired up with their homologous pair. So you have 23 homologous pairs. Another term is haploid and diploid. So diploid, we use 2N to represent diploid. So 2N, diploid mean the same thing. It contains homologous chromosome, and it is the full set of chromosomes. So in humans, for example, 46 is 2N in humans. Haploid, we just write as N, and that is haploid, which would be 23 in humans. So one copy of each chromosome. So it's not homologous because you just have the one. So here again, another picture, one copy of each, that's haploid. When you have the two homologous pairs, that's diploid. All right, I'm moving on. Pause if you need. So in the case of sex cells, sperm and egg, right? We have our little sperm here with the tail. We have our egg. These are haploid. These are haploid. All right, and when the sperm fertilizes the egg, we get 46 chromosome and a zygote. So this is now diploid. Okay, and eventually this zygote will become an embryo, a fetus, and then a baby. Looking at the stages. So interphase, we know interphase, this occurs before meiosis 1. And again, just like in mitosis, this is where DNA replication occurs. So breaking this down here, we have meiosis 1 at the top, and then if you scroll down further, then we have meiosis 2. So let's just take this one at a time here. So couple things happen here. Prophase 1, which comes after interphase, this is where pairs of homologous chromosomes come together, okay, and form a tetrad. Crossing over can occur, and so this is a main event that occurs, a very important event. And over here off on the right, I have crossing over. So what you see happens is you have this homologous pair, and their little chromatids touch and they swap genes. So what has happened is some of the stuff, characteristics from the one chromosome is swapped with another. What does this do, or what's the point of that? Well, this is where genetic diversity comes in. So it creates genetic diversity. And this is a random thing that occurs. So very important, major thing that occurs. And that's why, for example, if you have siblings, that you look different than your brothers or sisters, all right? Because this has occurred during this process of meiosis to the sperm and the egg. All right, metaphase 1. Again, in metaphase, you know they line up in the middle. Anaphase 1, chromosomes separate. Lastly, you have two new nuclei and the nuclear membrane reappears. So this kind of looks like mitosis, right? So now we continue. We leave off from telophase 1 and we go into meiosis 2. So you'll notice I have these little ends across here in green. These are now haploid cells. And again, the nuclear membrane breaks down, the spindle fibers form. Metaphase 2 line up in the middle, then they pull apart. And now you have 4 new nuclei that are genetically different. So all 4 of these are genetically different because of that crossing over that took place during meiosis 1. All right. And that's it.